So these are what we call cells. We grow them in a flask like this, and they usually
Today, I'm going to share with you what I actually do in the lab as a cancer researcher. So before we start, we first need to make sure we are protecting ourselves as well as the cells. So first you need to make sure you're wearing a lab gloves, and you need to make sure you're wearing properly attired in a lab coat. So to start, we have this machine here, which is called an incubator. So basically, this is where the cells actually live. So they are kept in this kind of plastic containers. So we are going to take a closer look in how they look. So as discussed in the slide, these are cells. So cells are basically kept in this kind of flask, and then the red solution on top of it is basically the food that we give to the cells so that they can grow and they will be happy and healthy, because cells get hungry too, just like us. But you are not able to see the individual cells. But look what happens when we bring this and show them under the microscope. So this is a microscope, and then if we put it and the cells over here, and then we zoom or we increase or make them bigger, and then you will be able to actually see the cell. What you see here, these are called cells, and as you can see, they are very tiny. So our body is actually made up of multiple of these, and they form our eye, our nose, our hands, our feet, our heart, and everything we have. So when they become unhappy, so that's when we fall sick, and then we need to take medicine to feel better. So because of that, we actually need to give them fresh food so that they actually will feel happy and they can grow, that we can use for our experiments. So we will actually put them into this we call this a hood or a system, and what we have in the red bottle, it's actually the food for the cells. So we will give that to the cells, and so that we can actually have them grow bigger and have more of them for our work. And once we have given the food in here, and then we will actually bring them to this and then place them in there. So this is the machine where they actually feel the most happiest to stay, and they will be healthy and they will be alive once we put them inside here. Okay, for my work, I actually put the cells that I have into a tube like this, and then I actually color them red, and then we will see later why I need to have them in a red color. Now before we do that, we actually will go and get the cells to the bottom of this tube. Over here we have another machine. So when you put these cells in the tube inside of this machine, and what's going to happen is they are going cells in this machine. What it does is it basically spins the sample, like goes in circles. So if you have played in a merry-go-round, so that's what actually happens. The cells are going to be circling like that. We have now taken the tube from the machine after spinning around, and if you can see at the bottom here, so can you see there's a very small white color part? So those are the cells. So these cells are now actually colored, and we are now going to bring this and put them in the fish, because that's how we do our work. So we now go to the other room where we will work. Okay, so once we have the cells prepared and colored red like this, we take them and we actually add them to the fish that are shown over here. So you would have seen the zebrafish larvae or the babies as before. So what we use is we use a needle like this, and then we add the cells into them. So what happens when we do this or inject it? With the cells, what we do is we use this microscope and we actually shine a white light. And if you shine a white light over here, what you can see is you can see the baby. So that's the eye. We have the heart over here, and these are the colored cells, which I colored them red, and you can see them over here. But look what happens when I shine a green color light instead. So when I have the fish in, this is what we can see. So we are going to now in my work see this red color, whether it grows big or if it grows small when I give them medicine. So after giving medicine for 3 days, you can actually see the fish or the baby fish has actually grown bigger, and you can see that the cells are no longer here, but they have actually moved. And if we look under the green light, what you see is actually the big red amount of cells is no longer there, and we have only small red dots. So this actually means that the medicine we gave them is actually working, and the fish is now happier. So this is basically what we do. We try to see how different medicine works so that the fish will feel better. I hope you enjoyed following me and being a researcher for a day. Bye.